Alrighty, and so our first presentation is going to be Duan, and she is going to be presenting over, excuse me, let me find my thing. So Sia is going to be presenting over how seizures affect learning and memory. And so if you could go ahead and she's also from Baylor University, as all of our presenters are in this group. So you can go ahead and share your screen. I've enabled that. And once you get started, I will give you the 12-minute timer, and I will give you those cues as you wind down on your presentation.
Sounds good. I'm looking for it right now.
Okay, no worries.
Okay. Yeah, so can everyone see my screen? Wait. Okay. So yeah, hi everyone, my name is Dawn, and today I'm going to present on a recent research project that our lab just conducted, and this talk is going to be titled Contextual Memory is Impaired for Up to 6 hours Following a Status Epilepticus. And so this is a brief overview of my talk, and first I'll be giving you an introduction into our research and why it is relevant today. And then I'll be showing you some literature review on past studies, just to introduce you to, common trends and patterns that have been found in the past. And then next, I'll be going into the specific methodology that we use for the study, as well as the results that we found, and then some conclusion and potential applications of the study. And so first, I want to talk about epilepsy, as this is one of the main focuses of our lab. So epilepsy is a chronic non-communicable disease of the brain, and epileptic individuals are characterized with recurrent seizures, loss of awareness or consciousness, disturbances in movement, sensation, mood, or cognitive functions. And then statistically, there are about 50 million people worldwide suffer from this condition. And unfortunately, a lot of these cases actually take place in third world countries. And so because of that, the individuals don't really get the adequate treatment they need. And then on top of that, they also suffer from a lot of discrimination and stigmatism from their own community. And then in America alone, there are about 3.4 million Americans diagnosed with epilepsy. And so now I'll just be going into the literature review on past studies. So overall, seizure frequency is associated with a lower quality of life. So the more often someone has seizures, the worse quality of life that they report. And then there have also been some studies done on animals, and these studies have generally found that multiple seizures often lead to memory impairment. And then recently, researchers have also been interested in the effect of a single seizure. And so this study by Pearson here found that learning is impaired for up to a week later after one seizure. However, there were also some limitations that we found with these studies, is that they really mostly focus on only the effects of multiple seizures. And so along with that, the period of post-seizure amnesia is not well researched. And with that being said, the goal that we have for the study was that we wanted to investigate the effect of a single status epilepticus, which is a seizure that lasts for more than 5 minutes, having 2 seizures within 5 minutes, and just see how that could affect learning and memory in mice models. And with that being said, this is the methodology that we use for our study. So we group mice by 3 main factors, and those were injection, training time, and then testing time. And so for the injection, we would first inject the mice with saline or canic acid, and the groups that were that received the saline injection were the control group, and the groups that received the canic acid were the groups that were induced seizures. And then 1 hour or 6 hours following that injection, the mice would then be put through training time, which I'll explain in the next couple of slides. And then lastly, 24 hours or a week later following that training time, the mice would then be put through testing time. And so overall, we had 8 groups in total. And so we had groups such as saline, 6 hours test, 1 week, 6 hour train, 1 week test, and then canic acid, 1 hour train, 1 week test, and etc. And so now I'll just be explaining what delayed field conditioning is and why we use this task as the main testing model for our study. And so delayed field conditioning is an associative learning task that trains mice to associate a conditioned stimulus with an unconditioned stimulus, which will hopefully result in a conditioned response. And in our study, if you look at this figure, our conditioned stimulus would be the sound that the mice would hear. And then the unconditioned stimulus is the electric shock that they would receive. And then lastly, the conditioned response would be the freezing that they responded to the electric shock. And then this is a frequently used model to study learning and memory in mice. And so with that being said, for training day 1, which happened an hour or 6 hours following that injection, the mice would be placed in a testing apparatus for a little more than 5 minutes. And during this time, the mice would first hear 2 periods of white noise sound, and followed by that, they would receive a slight electric shock. And the point of this task is just to train the mice to associate that experience of being in the testing apparatus and hearing that sound to the electric shock that they receive. Then next, in the context test, which took place 24-hour a week following that training time, the mice was then placed back into the same testing apparatus. However, this time there was no electric shock. The mice was simply put in that testing apparatus for a little more than 5 minutes again. And the point of this test is just to test the mice's ability to recall that previous experience that they received of being back in the same testing place, and just recalling that shock that they got on training day, but this time without the cue of being the sound. And then lastly, in the cue recall test, which was 2 hours after context test, the mice was then placed in a new testing apparatus. And to make this possible, we actually change up as much of the testing apparatus as possible. So we place vanilla scents underneath the testing apparatus. We turn off lights. We use paper towels in transfer cages, etc., just to really give that mice a new testing experience. And then following that, the mice were then placed in this testing apparatus again. And this time they would hear a period of sound. However, this time there was no electric shock. And the point of this is just to test the ability to, once again, connect that experience of hearing that sound and connect it to the shock, even though this time they were placed in a new testing environment. And through all these 3 tests, we actually measure percent freezing, which is a measure that we use to test the mice's ability to recall their context. So if the mice show higher freezing percentage, that means that they were able to better recall their experience, and thus they had less memory impairment. And so this is the result that we found in a context test, which again was the test when they were placed back in the same testing apparatus without the sound. And we found that overall, status epilepticus reduces context memory. So if you look at this graph, the saline groups show significantly higher freezing percentage compared to the canic acid groups, and which show that their memory was less impaired and they were not as affected, or they were better able to recall the experience that they had. And then we also found an effect of training time in the mice recall ability. So in looking specifically at the canic acid groups, the groups that were trained 6 hours following their injection perform higher freezing percentage compared to the groups that were trained an hour after. And this showed that when they were injected 6 hours, when they were trained 6 hours following the injection, they were able to better recall the experience later on. And then next, in the cue memory test, which again is the test when they were placed back in the new testing apparatus and with the sound, but no electric shock. And this time, interestingly, we found no effects of seizures on cue memory. And so regardless of if the mice had seizures or not, their cue memory was affected the same way. And then we again found an effective training time on their recall ability. So groups that were trained 6 hours after their injection perform better than groups that were trained an hour after. So you look at this graph again, the canic acid groups that were trained 6 hours following that injection show higher freezing percentage compared to groups that were trained an hour after. And so with that being said, the conclusion that we have for this is that overall, learning is more impaired an hour after a status epileptic event compared to 6 hours after. And then additionally, contextual memory appears to be particularly susceptible to alterations, as the results we found in the context test was much more robust compared to the key memory test. And so some potential applications that this study could have is that could be used to inform patients of possible learning and memory limitations following an epileptic seizure. And so specifically the time frame that we use, so like the 1 hour, the 6 hour, or even the 24 hour and the 1 week could be used to, investigate ways and find ways that could help epileptic patients best learn following their epileptic episode. And then additionally, it could also guide future research into quality of life interventions following a single seizure. And then we could also look at how seizures affected the brain at a molecular level and some interventions that we could have from that. And for our lab specifically, one future direction that we could take is that we could perhaps use rapamycin to suppress mTOR signaling in the brain following a seizure. And this also help us to possibly retrieve some of the memory loss in the mice. And so with that being said, I want to express special thanks to my lab for allowing me to be a part of this project, as well as my PI, Dr. Lugo, and graduate student Greg Solins for helping me put together this presentation. Thank you.
Awesome, thank you. Now is the time for some judge questions, and then we can go with our audience questions if there are any.
All right. Thank you so much. That was nicely presented. I have a few questions. So do you think that these results would extend to non-stress related memory impairments? Because with fear learning, it's like electric shock, these sorts of things. But other sorts of behavioral tests that could be done could be like maze learning or social or like object recognition, like memory sort of tasks, or even like reward learning. So something that's like non-stressful related. So what do you think would happen in these animals?
That is a good question. I know they also did some motor tests outside of this, but I don't have the results with me right now. But I feel like without stress, the mice would react differently, just because I feel like in general, just we also react differently without stress or with stress, and so that would be really interesting to look at further and see how, like, without the electric shock, maybe would they be able to better recall their experience, or like if they would be able to associate that experience with with everything. But yeah, that would be interesting to look at.
Yeah, especially because your effects were so pronounced with the contextual and not with the cued memory, right? Another question is, could you tell me what the age of these mice were? And do you think that age is a large factor, especially since maybe some epileptic, impairments may start really young?
Yeah, so these mice were 2 months old. And so since they were 2 months old, that means that they were fully grown and developed mice. And so how that could translate to, perhaps human models would just be that that could be adults models, or like even like older children. So yeah.
Perfect. All right. Thank you so much.
Thank you.
All right, thank you. You can go ahead and unshare your screen, and yeah, just thank you for the presentation. And so next up we have Catherine, also from Baylor University, who is going to be presenting over defective mitolyso contact sites in mitochondrial diseases. So the time is yours. Go ahead and start, and I'll start my timer as soon as you start speaking.
Thank you. Hey. Good morning. My name is Catherine Molkel. I'm a student at Baylor University. Today, I'm going to be talking on defective mitochondrial lysosome contact sites and its dynamics in mitochondrial associated diseases. And this research is part of Dr. Wang's lab, which is a neuro-based lab at Northwestern Feinberg School of Medicine. So our lab has previously studied and shown that there is crosstalk between mitochondria and lysosomes, which is mitochondria and lysosome contact sites dynamically form. And this slide goes over the 3 different images used to show the formation of mito and lyso contact sites. So first we have live SIM. SIM stands for structured illumination microscopy. We have a mitochondria, which is labeled by M Apple Tom 20, which is the red color. Then we have lysosomes labeled by LAMP1 GFP, which would be the green fluorescent protein. As we see in this image, there's a contact site formed between the mitochondria and lysosomes. Then we move on to 3D SIM and we see contact site being formed in 3 different planes. And lastly, with the live cell confocal imaging, we see the entire process of the contact site being formed and separation. So at 0 seconds, we see the lysosome and mitochondria are not in contact with each other. At 8 seconds, a contact is formed between the lysosome and mitochondria, and it stays together with each other throughout 68 seconds. Finally, at 74 seconds, it finally separates from each other. So the contact site finally separates, it finally comes untethered. So we go into the next slide. Our lab has shown that mitochondria and lysosome contact sites play a significant pathway in neurodiseases that I've been studying, such as Parkinson's, Alzheimer's, and also cellular metabolism. But in the basics for today's project, mitochondria and lysosome contact sites regulate organelle dynamics. So that would be the bidirectional regulation of both the mitochondria and lysosomes. So that would be the modulation of organelle network dynamics. And I've got an image here to explain, for example, clustering. So this image has been taken from a review paper where I was co-author with my lab. We have clustered mitochondria and we have clustered lysosomes. And then a contact site forms between the mitochondria and lysosomes. And when the contact site finally separates between the mitochondria and lysosome, we see the cluster of mitochondria finally unclusters, as well as the clustered lysosome unclusters. So this is how the organelle, the contact site leads to bidirectional regulation. There's also metabolite transfer at these sites, so that would be transfer of calcium, iron, and cholesterol. As we go into mitochondrial DNA, it is DNA found in the mitochondria. It is double-stranded with 37 genes. 13 of these genes are for oxidative phosphorylation of ATP. So I've included the image of the whole ATP process. And as we can see, all the colored proteins in the intermembrane space that contributes in the phosphorylation, they're all part of mitochondrial DNA. Then we have 22 genes that provide instructions to make tRNA, which is transfer RNA, and 2 genes to make rRNA, so ribosomal RNA. And as we get into POLG2, POLG2 is located in the mitochondrial matrix. It forms a heterotrimer with 1 catalytic subunit POLG and 2 processative subunits POLG2. So as I've mentioned before, this is the heterotrimer that is formed. We have 1 catalytic subunit, this is POLG, and we have 2 copies of POLG2, which serves as a processing subunit. So this is the heterotrimer in the mitochondrial matrix. So what would be the function of POLG2? POLG2 is responsible for mitochondria DNA replication. So we get 2 mitochondria DNA from the whole replication process. And as I mentioned before, POLG2 forms the processing subunit of the mitochondrial DNA polymerase gamma. So these are the 2 mutations that I used for my research project for today. The first mutation is P205R, which is an autosomal dominant mutant. It's seen in this circle right here. This mutant leads to disease in humans such as lethargy, central hypotonia, which is weakness, decreased muscle tone, liver disease. And then the second mutation was R369G, which is also an autosomal dominant mutant disease, as seen in this circle. And this leads to diseases such as external ophthalmoplegia, which is weakness in the eyes. We have AC fatigue, muscle weakness, lactic acidosis, which would be buildup of acid in the body. So while these mutations cause diseases in humans, it also leads to defects in the mitochondrial DNA. Since now the mitochondrial DNA is not able to fully replicate properly, the processing subunit is affected and can work as well. And now we can jump into my research question. So my research question was, how do mutations that disrupt mitochondrial DNA affect the mito and lyso contacts. So how did the mutations that I just mentioned, P205R and R369G, how did these mutations that disrupt mitochondrial DNA affect the formation of mito and lyso contact sites? So I studied 3 different conditions, the wild type, P205R, the first mutant, and the second R369G. And the 2 channels that I primarily focused on was the mitochondrial channel, which is again labeled by M Apple Tom 20, and the lysosomal channel labeled by LAMP1 MGFP, the green fluorescent protein, and this is the red color protein. And my analysis focused on the effect of POLG2 disease mutations on mito and lyso contact pairing. It also focused on the effect of these mutations on mitochondrial dynamics, in specifics, the motility, and lastly, lysosomal dynamics, in specifics, motility and clustering. I hypothesized that the POLG2 mutations would disrupt contact pairing between the mitochondria and lysosomes. POLG2 mutations would affect mitochondrial dynamics by causing slower mitochondrial motility. And lastly, no effect of POLG2 mutations would be seen on lysosomal dynamics since it is more based in mitochondria. And as we go into my first result, POLG2 disease mutant disrupts contact pairing. So first I looked at the amount of lysosomes in mito and lyso contact sites. So that means how many mito and lyso contact sites were formed across all conditions, and they were all around the same. There wasn't any significant difference. So then we looked into mito and lyso minimal contact duration time. This basically means we have a mitochondria, we have a lysosome, and they come in contact. So what is the minimum duration time that the mito and lyso stays in contact before it finally separates? And when we did the analysis, I did notice POLG2 R369G did have longer minimal duration time in comparison to the wild type and the first condition P205R. And this was significant, and we wondered if this difference was caused because of the mutations effect on mitochondria or the mutations effect on lysosomes. So as we go into that, we looked at the POLG2 mutations on mitochondrial motility. I just included an image of what I was analyzing. So at 0 minutes, I would track a specific mitochondria and follow it through the whole 3-minute frame. I would look at the speed, the distance, as well as the shape of the mitochondria. So when I did the analysis, I didn't notice any significant difference in the mitochondrial motility in any of the 3 conditions. We then plotted this data we plotted each individual mitochondrial motility point in the graph, and they were all around the same area, so no significant difference was seen of the mutations on mitochondrial motility. So then we looked into how it affected lysosomal motility. Again, at 0 minutes, I tracked a specific lysosome, and then throughout the entire 3 minutes, the same lysosome, I would look at the speed, distance, and shape. And when I did analysis again, no significant difference of the mutations were seen on lysosomal motility in any of the 3 conditions. We also individually plotted the data, and they were all around the same area in the graph, so no difference was seen. However, when I was doing the analysis, I did notice more lysosomal clusters were formed in the mutant conditions, so we looked into that. And this is what I looked at. So this is the lysosomal channel, and we took a cluster to be 2 or more lysosomes attached to each other. So this would be a cluster of lysosomes, that would be another lysosomal cluster. So when we plotted the data, there was significant difference. POLG2 P205R had more clusters formed at around 2 to 3 per experimental video, and POLG2 R369G had even more clusters formed in comparison to the wild type and P205R. So this was significant and interesting to see, since POLG2 is mitochondrial based, but it somewhat affected the lysosomal dynamics, especially the clustering aspect. So to conclude, the effect of POLG2 disease associated mutations, POLG2 mutation R369G does disrupt mito and lyso contact pairing, since it leads to a longer minimal duration time of the contacts being with each other. There was no effect of POLG2 mutation seen on mitochondrial dynamics, and that was really interesting since POLG2 is a mitochondrial based mutant. And POLG2 mutations P205R and R369G do disrupt lysosomal dynamics by affecting the cluster of lysosomes. As I went before, it led to an increase of clusters formed in the conditions. And currently I'm still trying to understand the effect of POLG2 mutations on the mito and lyso contact dynamics. So I'm looking at how it affects and disrupts density within a cell. And this also sparked future topics of research. I would like to know what elements of the POLG2 plays a role in lysosomal regulation at the contact sites. And also, can POLG2 mutations play a role in neurodiseases, especially Parkinson's? I've been studying that disease, and it was morally affected by the failure of lysosomal regulation at contact sites. I want to see if POLG2 mutations could have a role in that. I would like to give acknowledgement to my lab, to Dr. Wong, who was my PI for helping me throughout this research project, and Jasmine, Eric, Taylor, and George, who also helped me with analysis and imaging of the cells. My lab is right there. It has a really pretty scene. And yeah, thank you.
Awesome, thank you. Great job. Now the judge questions time.
All right. Yes, thank you so much. Very professional. And congrats on being a co-author on that nice review. Okay, so I am wondering a little bit about your methods. Were these conducted in mice, these types of experiments, or cells, or how did that work out before you did the high level microscopy?
So these were conducted on HeLa cells. So Jasmine, she's my lab manager. She would inject like the different conditions, the HeLa cells, transfect them, and then we used the two microscopy we used were Zeiss microscopy and Nikon live cell microscopy. So that's how we study the cells.
Great. Okay. So I'm wondering, since these were done in cells, what would be, do you expect that the translational validity would still hold up like in mice, in humans, for example?
I think it is hard to say in humans per se, but like I feel like that we would still see around the same effects because these were done in HeLa cells and they're also very similar to human cells and animal cells. So there would be around the same effects, but I couldn't say it would be the exact same.
Okay, great. And then I'm kind of interested in the transmission between the lysosomes and mitochondria. So you said that some of the messengers would be like calcium, iron, cholesterol. So do you know, I mean, this is a very specific question, but do you know if there is any, like if you give them more neurotransmitters, if their contacts like become closer or spread apart, like how have there been experiments to kind of manipulate their messenger systems?
Are you referring to, sorry, are you referring to the metabolic transferase messengers?
Yeah, just any if these messengers, like if you put more calcium or more iron, like do the contacts form more closely?
That is a really interesting project. And I think in a review, we did the review paper that I worked with my lab, we did notice that when we increased the amount of calcium and iron, they weren't able to regulate itself. Like it did form problems with the contact sites. It actually stayed longer in duration in the contact sites, and it was harder to like separate them both. Oh, I think you're muted.
Oh, great. Yes, thank you so much. That was very interesting.
Thank you.
Alrighty, thank you so much. We're going to, excuse me, sorry. We're going to move on to the next presentation, and that will be with Pranay and Sindhuja, who are also from Baylor, and they are going to be presenting on the effect of chronic mild stress on cisplatin-induced affective syndromes. And so you guys can go ahead and share your screen, and I will start recording with the 12 minutes as soon as y'all start speaking.
Can all y'all see that? Sweet. All right. Cindy, are you ready?
Yeah.
Sweet. Okay. So hello, my name is Pranay, and my partner's name is Sindhuja. We're both lab members at the Psychoneuroimmunology lab here at Baylor, headed by Dr. Rachaya. And the project that we're going to be talking about today is exploring the effects of chronic, unpredictable mild stress on cisplatin-induced affective symptoms. So nearly 1.6 million people are diagnosed with cancer every year. The diagnosis and prognosis of cancer can be a very stressful event for many individuals. And then on top of that, a lot of patients experience chronic stress, such as the treatment process of cancer, as well as different financial burdens, and even just the diagnosis itself can be a very stressful event for many people. We currently know that there's very burdensome experiences from these stressors, and we were looking to see how these chronic mild stressors could impact overall cisplatin affective symptoms. The current study was exploring how these stressors would impact the affective symptoms of cisplatin.
So in this study, we used a stressor model called the unpredicted chronic mild stress. And this is a protocol that's typically used to induce chronic stress in mice. It is a rodent model for depression where the rodent will experience multiple different stressors over a given period of time. So some examples of these chronic stressors included a tilted cage, predator odor, restraint, strobe light, and no bedding in these cages. And so these stressors were limited to a couple of hours in this study and were designed to mimic like the hassles of daily lives that people typically experience. So the first known model of UCMS was actually introduced in 1982 by Katz, but since then it's been developed by multiple researchers, including P. Wilner, and most recently in 2017. Another important aspect of this study was the use of cisplatin, which is a first-line chemotherapy drug that's typically used to treat various cancers, including head and neck cancer. It's typically administered through a vein or IV as an infusion, and mice typically respond to cisplatin treatment very similar to humans. So it's often used in murine models to serve as a chemotherapy treatment and to observe its side effects.
So with that, we believe that unpredictable chronic mild stress would exacerbate the cisplatin and depressive-like symptoms. And so the way we went about to conduct this experiment was using 30 different mice. We did a 2 factorial design in which we split up the mice into the chronic mild stress group and then the control. And then after that, we either injected the groups with cisplatin or saline, so that roughly left around 7 to 8 mice per group for 4 different groups. And so kind of like the timeline of how everything, how we went about doing this was the first week, day 0 to day 7 was handling, which is where we allow the rodent to get accustomed to human interactions. After that, we started the UCMS protocol starting day 9 for 2 weeks. And so in this model, we did 2 different stressors every single day, one in the morning and one in the afternoon for 2 weeks straight. And so some of the examples, like Sindhuja said, there's no bedding, wet bedding, food and water restriction, the strobe lights, there's a light cycle disruption, predator odor, restraint, and tilted cage. After 2 weeks of this, we started injections, which were 5.28 milligrams per kilograms of cisplatin. And then if the mice were part of the saline group, they were given the equivalent volume as well. After 3 weeks of that, once every single week, we conducted 2 different behavioral tests, novelty suppressed feeding, as well as tail suspension test, and then collected tissue. One thing to be noted in this diagram, we did also do LMA or locomotor activity starting day 25. It was used more as a way to gauge the rodents' overall health and making sure that they weren't having any adverse effects from the cisplatin or saline.
So body weights were also collected throughout the study. And with these body weights, we do see, as expected, that the cisplatin mice have lower body weights than the saline mice. And we also see that the mice that underwent the stressors in the UCMS group receive saline have higher body weights than the other groups of mice. We also noticed a significant stress cisplatin and time effect, where the UCMS cis significantly decreased in body weight throughout the study. And just to note that this graph is from the beginning of the treatment itself and not the beginning of the UCMS. And so this is also a graph of the locomotor activity that Pranay was talking about. So during this task, mice are basically placed in an empty cage, and the distance that they traveled is quantified using NOLDIS, which is a video tracking software. And we use it as a simple assessment of general mice health. And so as the mice become more accustomed to this environment, they show a decline in activity. Therefore, an effect of time was observed. So we also see an effect for cisplatin, where those that are undergoing treatment have less movement. And the control cis group here also shows a greater decline on days 2, 9, and 16. And these are the days after cisplatin treatment, while showing a recovery on the next few days. And this effect also approaches significance.
So the next behavioral task that we did was novelty suppressed feeding. There's two different aspects of this. First, the rodent is placed in a novel environment, and a food particle is placed in the middle of the open field, and the amount of time it takes for the rodent to eat the food is tracked. That's another way we measure that as latency. And then afterwards, we place the rodent back into the home cage and see how long it takes for them to eat that food, just to make sure that the reason why they're taking so long to eat the food in the novel environment was more because of anxiety-like symptoms and not because of a lack of hunger. Generally, we tend to see an increased latency to eat is associated with a lot of mood and anxiety-like symptoms. And so when we conducted this in our experiment, we observed a really robust effect on the stress feeding behavior, but there really wasn't anything or any interaction with cisplatin, which was kind of surprising.
So the next test that we conducted is the tail suspension test, which is sensitive to antidepressant activity. So during this test, the mice are hung by their tail, and the time that they're immobile is recorded. So we see here that we have an interaction between stress and cisplatin. So the UCMS mice that received cisplatin moved more than those that received saline. This is typically not what we would expect, because depression is usually associated with increased immobility. So it is possible that the cisplatin induced neuropathy within the tail, which then caused the mice to experience greater discomfort during this task, which caused them to move more and become more mobile. So however, more research will be necessary to investigate this possibility.
So we ended up collecting tissue, and we found that the spleen weight was significantly impacted by stress. It's well known that the immunomodulatory impacts of stress can impact spleen weight, but the direction and the modulation is dependent on the type and duration of the stressor. So when we take all these pieces of data together, the stress paradigm that we utilized did seem to have it induced the physiological response that we were expecting. But when we were collecting the adrenal glands, we did not find any differences. And so that could be due to various factors, including the fact that it's harder to see the adrenal weights change, especially after having a huge gap between the stressor and tissue collection. If you remember back to the slide of the timeline, there was almost 20 days between when we stopped the stress model and collected tissue. So this is a lot of time where visually you might not see any differences in the adrenal glands. Another thing that we noticed while collecting the tissue, there was a lot of adipose tissue surrounding the adrenal glands. But once again, this is something that we have to explore in the future, and that might be something that a future study looks at.
And so overall, cisplatin has shown to have a negative effect on body weight and metabolic activity. This is something that we would expect because previous studies have found that cisplatin administration results in significantly decreased feeding efficiency, elevated renal oxidative stress, and reduced antioxidative activity. UCMS prior to cisplatin did not result in more severe depressive-like behavioral. It instead did increase body weight and novelty suppressed feeding. And this is expected because cortisol, which is our stress hormone, stimulates fat and carbohydrate metabolism, which creates a surge of energy in our body. So this in turn increases one's appetite, which results in an increase in body weight under stressful conditions. UCMS was also shown to decrease in spleen weight, which is possibly due to the immunomodulatory effects that Pranay was talking about. And so overall, based on our findings, UCMS may have a role in enhancing cisplatin-related fatigue. However, future research would be needed to support this. In the future, we could also look at specific ways to evaluate fatigue-like behavior specifically. And this study should also be done in female mice because we only use male mice in this. And we are currently also using this model again in tumor-bearing mice to see whether stress will exacerbate immunological function with specific LLC tumors.
We wanted to give thanks to Dr. Vichaya, who is our head PI, helped to put this presentation together as well as the study, and two grad students, Haley and Caleb, both who helped throughout this entire process, and fellow undergraduate researcher, Colin. All of them helped to make this possible. Thank you.
Awesome. Thank you all. Now it's the judge question time.
All right, nice job, guys. I'm impressed that you had some really nice stress effects on your behavior. You know, I think that it is difficult to find, but if the protocol works well, then it's nice to see, let's say. Related to that, so the unpredictable chronic mild stress is one certain stress protocol, but do you think that using other types of stressors combined with cisplatin would yield similar results?
I would think so. I think the reason why we chose UCMS, I think, was its effects are very robust, and there's a lot of literature on it, and I think that's kind of why we decided to use it. But I would 100% think that if we used another stress or another stress model, we would see at least some similar results to what we saw.
Nice. Yeah, I think that because it has pretty high translational validity to our human, lives, yeah, great. So about the cisplatin, I like how you explained that it was used for certain types of cancers for treatment. I'm wondering if you can tell me why you chose the dose that you did.
So the dose for cisplatin is typically going to be subjective and kind of relevant according to everyone's body weight. Even in humans, they actually measure height, body weight, and relative adipose tissue as well, because sometimes since it is administered through a vein, if it can cause severe tissue damage if not given the proper dosage. So this is kind of a standard protocol that we use. Typically in mice, I think we take the average of their body weight and then relate it back to that. So we do mg per kg for that.
And to add on to that as well, previous study, we've done different concentrations of cisplatin before, but we've seen that mice die off way too quick. And so that was kind of the reason why we ended up with this dose.
Great. Good. Okay. I was going to ask why you thought there was an increase in body weight after stress, but you did address that in your discussion. But I'm as my last question, kind of wondering about what you think future directions would be for figuring out this fatigue that they potentially have, and pain, like you said, like maybe hyperalgesia, like follow-ups with this, since you saw like very profound differences from the tail suspension test and the novelty suppression of feeding.
Yeah, I think kind of doing more research on the different types of behavioral tasks there are would help a lot, because obviously there are very limited things that we can do in the lab versus like the number of protocols that are out there. So kind of researching more on specific behaviors that isolate that fatigue like phenotype specifically would be very helpful, because right now it's hard to tell whether the mice was already previously stressed out or whether they were getting stressed out through the UCMS model. It's kind of hard to gauge that. So kind of just doing more research and figuring out which behavioral test kind of identifies and isolates that phenotype the best, I think would be the best way to go about it.
Great. Thanks, guys. Keep it up.
Thank you.
Thank you.
All right, so this concludes the Neuroscience 2 division of the HOT Research Conference. And so we're going to do a picture now. So I'm going to go ahead and stop the recording.